Chapter 15, Nonspecific Lines of Defense. This is going to move us into the immune chapter, Chapter 16. We're going to be spending a couple of weeks on that chapter. Actually, there's 3 chapters. First, let me introduce you to a really important point. Nonspecific versus specific resistance. The view you're looking at here is showing you microorganisms moving through the skin, first line of defense, as well as mucous membranes, breaks in those areas. If they get past and into underlying tissue, they run into the second lines of defense, phagocytosis, white blood cells, inflammation, a variety of other things. If they get past those, most of the time first and second lines of defense keep organisms down. But if they get through, then your immune system kicks in, and that is called specific resistance. So nonspecific resistance it's geared towards combating anything. It could be a worm, a virus, a bacterium. It could be tattoo ink. It could be a sliver of glass. It could be a wood splinter. Doesn't matter what it is. It could be something alive. It may not be anything alive. Specific resistance is geared towards a specific thing, an antigen, I'll call it. Once your immune system has responded, it recognizes that antigen, that object that it reacted against, and it will produce a quicker response the second time, third time, and so on. Again, we're going to have a couple of chapters on specific resistance, so you're not going to hear about this again in the rest of the chapter. We're only talking non-specific resistance. So again, specific resistance is the same thing as immunity. All right, here's a look at some non-specific resistance factors. You can see they vary a lot. Again, first line of defense, skin and mucous membranes. Barrier, in other words, a physical barrier. But in addition to that, we've got all sorts of other lines of defense. Chemicals like mucus, saliva has lysozyme in it, urine has lysozyme in it. Urine also flushes the urethra out, keeps organisms down that way. You have cilia along your various parts of your respiratory tract. Skin is rather acidic. That helps to keep microorganisms down, as well as the salt content of skin. Then you've got some specific antimicrobial substances in your body. Their main job is antimicrobial. Interferon, which we talked about related to viruses, and we'll talk about complement. And then we're going to finish up with phagocytosis, inflammation, and fever. So again, these are all examples of nonspecific resistance. Okay, I've already introduced you to the first line of defense. I don't think there's much else to say on that. Do want to point out mucous membranes, which basically are skin inside out, or outside in, I suppose. All along your mucous membranes, which includes digestive system, respiratory system, genitourinary system, because there's some mucus glands there, you have got an antibody that is associated with mucus, as well as some other secretions of the body. It's IgA. Now, you might think that IgA, that this is specific resistance, but it's a very general antibody that really just kind of protects anything that comes in along a mucous membrane, like a respiratory tract. So if a flu virus enters in, you inhale it, IgA will attach to it and make it easier for a phagocytic cell to destroy the virus. It is not specific against a particular virus, a particular bacterium. So it's associated with secretions, and they're general protective antibodies. Okay, each of the organ systems has its own particular feature that protects it. So in the digestive system, you've got acidity. I've already mentioned lysozyme, an antibody. And then you've got a lot of digestive enzymes that break down protein, for example. And remember, bacterial structure and viral structures are mainly protein, as are most cells. They're enzymes or protein that inactivates metabolism, structural proteins, and so on. So remember pepsin in the stomach, various protein digesting enzymes that come from the pancreas, the small intestine. So you got a lot of them. Respiratory system, some key things. Tonsils, which are lymphoid tissue. You got a lot of white blood cells, including phagocytic cells there. You got cilia. You've got some general phagocytic cells in the lung called alveolar macrophages. Urinary system, I've already mentioned the flush of urine. Urine also has lysozyme. Again, lysozyme is a general antibacterial chemical. And reproductive system, things like vaginal acidity, hormonal variations that affect the lining, and so on. So, we've already talked about this. How do normal flora of yours protect you? They do not go on the offensive to kill other organisms. However, they take up space. You are a prime piece of landscape, and you've got lots of spaces all over your body, in and outside. And if these spaces or niches are occupied by your normal flora, there's no place for a pathogen or another organism could come in and set up house. So they take up space. That's their prime benefit for you. Just to define probiotics, but I'm actually going to cover this in lecture. I'm not going to cover this now. Probiotics are organisms associated with foods. The aim for their use is to rebalance your normal flora and take you back to a healthy state. So you probably would have best seen a probiotic associated with yogurt, which you see at the bottom right, Dannon yogurt, Activa, anything that has live cultures. But again, we'll talk more about this in lecture. The two key probiotic organisms are Lactobacillus and Bifidobacterium. Okay, second lines of defense, white blood cells. You're going to hear a lot about lymphocytes at the bottom right. All of these guys come from bone marrow. But for right now, key in on the neutrophils and a monocyte. He's not labeled here, but let me point to him. This guy right here is a monocyte, and the neutrophil and he are the key phagocytic cells in your blood. Now, these monocytes will move from blood out into the tissues, and when they move into the tissues, they change into what are called macrophages. Macrophages are out in the tissues. That's where they reside. Lung, liver, the dermis of the skin, lymph nodes, and so on. All the rest of these cells are found in blood, the other phagocytic cells like neutrophils and monocytes. Okay, I've introduced you to the white cells, and you see a blood vessel over here at the top with the different kinds. So let me point them out again. First, you got a red blood cell here. You got a platelet here, so forget those. They're not white blood cells. Okay, let me start over here with a neutrophil, who is pretty much in the center, major phagocytic cell. He's got two kind of sister cells. They're all related, these two guys on either side. On the left is a basophil. On the right is an eosinophil. Eosinophils are mainly involved with parasitic infections like protozoa and worms in particular. They detoxify, they remove toxins and poisons. They have some association with allergies. The guys on the left, basophils, a couple of functions. Number one, also associated with allergic reactions in that they produce histamine, which also is a mediator in the inflammatory reaction, so you'll see him later. And basophils also produce heparin, anticoagulant. And then farther out here, we've got a monocyte, the other professional phagocytic cell. again. Neutrophil and monocyte are the two key guys. And then the lone man on the right is a lymphocyte, and again, we will talk almost exclusively about him when we get into the chapters on specific resistance. So I've taken you through the white blood cells. By the way, the basophils have a very close sister cell in the dermis. They're called mast cells. When they're in blood, they're called basophils. The NK cells are a type of lymphocyte. They're generalists. They are not specific. They make no antibody. They mount no immune response. And we'll talk a little bit about them in upcoming chapter. Okay, out in the tissues, the two key phagocytic cells, macrophages and dendritic cells. The picture that was at the beginning of this PowerPoint, you might want to go back there now, is a dendritic cell. They have spiky little cytoplasmic extensions that go out in all directions. The guys that you see on these pictures, here is a macrophage, and this guy in a scanning EM is a macrophage. They're both phagocytosing. This one on the right bottom is kind of interesting because it's just chock full of bacteria that it's phagocytosed. Now the guys that are in the blood, for purposes of phagocytosis, they have to move out of the blood and go out into the tissues. So if there's inflammation out here, the phagocytic cells have to be able to get out there. Now remember, macrophages and dendritic cells may already be out there because they may already be in this tissue. Notice the blood vessel. Notice simple squamous epithelial tissue. This is a capillary you're looking at. Here is the outline of one of those cells right there. That is one squamous cell. Here is another squamous cell. Here is a third squamous cell. So these guys are thin in structure, flat, squamous, and simple, meaning there's only one layer, which makes it very handy for things to go through the wall. So white blood cells, like a neutrophil, can't actually go right through the middle of the wall. It's not like you could go through a wall of a house. But what they will do instead is, let me change colors here, is they will move out through the gaps between the cells and move into the area of injury. And that movement out in through the vessel wall is called diapedesis. It's the squeezing of the white blood cells through the wall. Now that obviously means that there is something out here that is attracting the white blood cell, because the white blood cell has no brain. It has no idea there's injury out there. So something must be being released to attract the white blood cells, and that process of attraction is called chemotaxis. Here's just a look at different places where you find macrophages and dendritic cells all around the body. All right, another second line of defense, phagocytosis. You've had this many times before, so I'm not going to belabor the point. 3 steps: entrapment, ingestion, and enzymatic digestion and kill. One important point about this entrapment: microorganisms that have a capsule around them are almost impossible to phagocytose. So there's a bacterium up here, this pink guy. Let me draw a capsule around him. Here's a capsule. It's a sticky, usually a polysaccharide, or sometimes polypeptide, layer around the outside of the cell. It makes it very difficult for a white blood cell to phagocytose it. That is the importance of entrapment. So because white blood cells can't trap encapsulated bacteria, your body has a plan, a gimmick, and that is general antibodies. You see them as little Y-shaped things right here. And their job is to coat the outside of the capsulated bacterium which then allows a white blood cell, here he is, to then now take in the entire bacterium. So capsules make it very difficult for entrapment, but opsonizing antibodies, or opsonin, can make it easier for the capsule to be coated and then trapped and ingested by a white blood cell. White blood cells can take in a lot of bacteria. If you look at the picture over here, you're looking at about 8 or 10 white blood cells. Here is one white blood cell right there. The nucleus is the dark blue round object, big one, and the little tiny blue things are bacteria that have been phagocytosed. I mean, they can be chock full of bacteria. And you see two scanning EMs here towards the left of white blood cells phagocytosing bacteria. And then you've got a picture over here. So enzymatic digestion and kill here's a better picture. So the microorganisms come into the cell inside of a vacuole, and they are now digested and killed inside of the white blood cell. And there are a lot of ways for the cell to do this. So there are a variety of enzymes you see inside the cell, lysozyme and glycolases and phospholipases and nucleases and things like that, that can erode the microorganism and break it down. Also, you see a superoxide radical right here, O2, wait a minute, that can be used to kill. Also, the cell can have H2O2 that is good at killing. There are peroxidases. So there's a lot of things going on inside of the white blood cell that contribute to killing. The picture you're looking at the bottom right is a white blood cell in blue phagocytosing a yeast cell in green, which is about the same size as the white blood cell. Okay, some chemicals. We're talking now about antimicrobial chemicals whose job is really antimicrobial function. And we're going to talk a lot about some of these chemicals later. They have a variety of names: cytokines, lymphokines, interleukins. They go by a variety of names. Some of these chemicals can act as poisons or toxins to destroy microorganisms. Some can enhance nonspecific resistance. In other words, some chemicals can turn on white blood cells to make them more effective at kill. So you see, whoops, I didn't mean to do that. There we go. Okay, so over in this diagram you see a picture of complement, which we're going to talk about in just a second. And notice that one of the things complement can do is it enhances white blood cells so that they're better killers. So look at those two white blood cells. Notice that they contribute to enhanced phagocytosis. That is, complement does. There are other chemicals. I mentioned lysozyme and tears. Again, we'll get back to complement in a second. Interferon, which is an antiviral chemical, alerts other cells to produce defensive chemicals against viral takeover when one cell releases interferon as a result of it being infected. Okay, so let's get to complement. I'm going to talk more about this in lecture. It's very involved. But basically, complement is a bunch of blood proteins. They're made by the liver. And these chemicals act as a chemical cascade of reactions. One reaction causing another reaction, causing another reaction, causing another reaction, ending with chemicals that contribute to the destruction of the antigen. So you see on the picture here, whoops, you see on the picture here that cascade that I was talking about. Look at this set of chemical reactions. right here. You can see quite a few chemical reactions. All right, so we start out over here with an antigen. Here's our bacterium. And the bacterium gets attached to antibody, those little purple Y-shaped molecules, with the addition of complement protein. Now this would be in the blood. So our antigen, our bacterium, is in the blood. Now, this complex of bacterium, antibody, and complement 1 causes the breakdown of complement 1, and it now changes to a different complement protein. That complement protein splits into a couple of different things. This protein now splits, when activated, into 2 more. This protein contributes to phagocytosis. The other protein then gets converted to another, which then activates a couple more complement proteins, 6 and 7, which, when activated, become complement 8, that activates a bunch of complement proteins all at the same time. These complement proteins, 5, a portion of 5, 6, 7, 8, 9, attack the outer wall structure of the bacterium, causing its lysis. That's the simplest I can explain complement. Along the way, notice that some of the complement proteins contribute to white cells moving out. Some of the complement proteins enhance the inflammatory reaction. So the end result here is lysis, destruction of the antigen. And again, another look at it. So again, look at the bottom of that sheet, that shot right there. Activation of the complement, a bunch of chemical reactions resulting in many more complement proteins being produced. Those complement proteins insert into the outer wall or structure of the antigen, and the antigen is then destroyed. That's the basis of complement. I've mentioned some of these terms already. These are called cytokines. Cytokines are just chemicals made by chemicals. They act as chemical messages between cells, and many of them are immune turn-on chemicals. Okay, so let's move over to inflammation. You've had this in A&P, if you've had A&P. We start out over here with a injury to tissue. In that case, it's a nail. Well, actually, in both cases, it's a nail. So we've gone through the skin, and we now have injured tissue right in this area or right in this area. And on the right-hand side, you also have the addition of bacteria that have come in. All right, now, remember, out here in the dermis, as well as over here in the dermis, are cells that are called mast cells. I mentioned them earlier. They are relatives of one of the white blood cells called basophils. They make histamine. So histamine is produced, and it accumulates out here. I'm going to put it in as red dots. Now, histamine has 2 major things that it does. It acts as a vasodilator. Blood vessels get larger, bringing more blood flow to the area. And histamine causes vessel permeability to increase. That is, the blood vessel becomes leaky or more porous so that things can move out of the blood and out into the tissue. And actually, you can see the advantage of that because that facilitates white blood cells coming out into the area. And of course, the increased blood flow, vasodilation, brings more blood and oxygen and nutrients and white blood cells to the area for repair. Now, there are other chemicals that have inflammatory effects. Prostaglandins, bradykinins, serotonin, but histamine is the main one. Okay, so I mentioned the two things that histamine does, and again, here they are right here. Permeability of the vessel wall and increased blood vessel diameter. So if you look at the picture down here, here's before inflammation, here's after inflammation. Look at the spaces in between the simple squamous cells. Now, obviously, white blood cells are going to squeeze out. That's what you see here. But look what else is going to come out. Fluid. What's the fluid called? Plasma. And what happens when plasma accumulates out here in the tissue? Edema. Swelling. That's from permeability changes. Now, over on the right, you see a blood vessel after inflammation. Let me go back to the first frame. Okay, here was the first frame on the left-hand side. This is right before inflammation has occurred. Now look at the second one here. Notice that the whole capillary bed has increased in size, bringing more blood flow to the area. So when you look at the area, it's going to appear red, and it's going to feel warm to the touch because of the heat being carried by blood. Alright, so what does all this give you? It gives you 4 symptoms. The 4 symptoms of inflammation: erythema, redness due to vasodilation; edema, swelling due to increased vessel permeability and plasma leakage; heat from more blood being carried to the area, sometimes called calor, C-A-L-O-R; and pain, sometimes also called dolor, D-O-L-O-R. The pain is from chemical release causing sensory neurons to fire, receptors to fire, as well as pressure from the plasma accumulation, edema, in the tissues. The picture that you're looking at the bottom is a case of hives. Basically, this is a mosquito bite times a million. So this is a good example of inflammation. I'm sure you've seen it before. Or you've seen inflammation before anyway. And again, another look at the process of inflammation. Okay, sometimes called the 5th symptom of inflammation, fever. It does not have to accompany inflammation. When you get a mosquito bite, you get an inflammatory response. There's no fever involved. Fever is generally associated with an infection, but it can sometimes also accompany inflammation. Now, don't confuse this with just localized heat. If you touch a mosquito bite that's undergone the inflammatory response, it feels warm to the touch, but the surrounding area is cool. Fever means increased heat. Your body temperature has been reset to a higher temperature. That's not the same thing as local heat. Okay, now in order for your body to reset temperature up, the area of the body that controls body temperature is the hypothalamus in the brain, and it can reset temperature upwards. And in order to get that body temperature up, you've got to generate heat, because it's not like you have a little furnace in your body like your apartment or your house does. So in order to generate heat, your muscles have to contract. You know that as shivering. So you're in a cold room, or you are getting a fever, you're in early stages of an infection, you start shaking. That's muscle contraction, and it generates heat, and your body core temperature increases. Pyrogens are the chemicals that can trigger the hypothalamus to reset body temperature. You can make your own pyrogens in the body. Many of your interleukins and cytokines are pyrogenic, but also bacteria can make pyrogens that do it too. When your fever breaks, you start sweating. That's how you release heat from the body. Now, there are benefits of fever, and I'm not talking about 105. That'll fry your brain and cause seizures. I'm talking about just a very slight fever of maybe 100, 101. You see a bunch of benefits here. All of these are proven effects of a low-grade fever. So very beneficial for both specific and particularly non-specific, both types of resistance. This is a diagram out of your book. I won't go over it. It just kind of encapsulates everything we've talked about, gives you a few extra things. The pictures you're looking at, the little girl's neck on the right is just showing you enlarged lymph nodes. That's called lymphadenopathy. And over on the right is a swollen leg. They've injected a radio-opaque dye into the lymphatic vessels to see where the swelling, what's happening, if there's an obstructed lymphatic vessel or something like that. And that is it. Short chapter.